Hello, my name is Andres Cisneros, and I'm a professor in the Department of Chemistry. And today I'll tell you a little bit about our research program and, in particular, one specific project. Before we start, I want to tell you that UNT, and me in particular, we're on Wichita land, so we're acknowledging the land, and Black Lives Matter. I also want to acknowledge all my group, current and past members, as well as collaborators, and funding from NIH, NSF, and NVIDIA, as well as computing time from UNT, previously Wayne State, and now Exceed and Microsoft. So first, I'm going to tell you a little bit about what my group does. We are a theoretical chemistry group. So what is theoretical chemistry? You can think about it as virtual chemistry. So we work on computers. So one of the computers that we use through Exceed is this supercomputer, which is the 5th largest in the world in an academic setting. It's called Stampede 2, and it's in the Texas Advanced Computing Center. The other one is Summit, which we have access to. This is the fastest supercomputer in the world. And so we can use theoretical chemistry to research several different things like new medicines, new materials, etc. We develop programs to run on these computers, and we use them for several different projects. I will tell you about one today. So how do we accomplish this? We use what are called molecular simulations. The aim of molecular simulations are to understand and predict molecular phenomena by calculating properties of microscopic systems. So we want to understand how these systems work by calculating energies and forces of the entire system. We can do this by using quantum mechanics, so the Schrödinger equation, or by classical molecular mechanics, using a force field, and we solve Newton's equations. By doing this, we're able to explore what is called the energy landscape, either the free energy or potential energy landscape, and this tells us properties of those systems. We apply these calculations for several different things. Today, I'm going to tell you a little bit about one particular application that we do for molecular simulations to investigate cancer mutations. And this started when we looked at DNA polymerases. So DNA polymerases are proteins that synthesize or replicate DNA. And so when DNA is being replicated, it's very important that these proteins do a faithful job of replicating DNA. So you don't want to have mistakes. Sometimes mistakes do happen. And so there are a lot of processes in the cell that are in charge of fixing DNA. And this is called DNA repair. So polymerases are involved in DNA replication. There are several different DNA polymerases. The replication of DNA is done really fast, so it takes 10 hours for 3 billion base pairs, and they're very, very good at doing this. So there's only 1 in 10 billion mistakes. So when we started looking at this, we used DNA polymerases as our first test bed for finding cancer mutations. Since then, we've gone on to explore other DNA repair and DNA modification enzymes. I'm going to tell you about 2 examples that are more recent. One was done by 2 students at UNT. One was Alice Walker, and the other one was Pavel Silvestrov, who found a novel mutation that is linked to prostate cancer in this enzyme called AlkB. So I'm going to tell you how we did this first. This project involves search of what are called single nucleotide polymorphisms, which are mistakes in the DNA in one single base that affect the final protein. So what happens in the cell in biology is you have the DNA that gets transcribed to RNA, and then that gets translated to a protein. And that's called the central dogma. If there's a mistake in the DNA, it can get translated all the way to the protein and have a mutant protein. So our initial hypothesis is if we have individuals that have a SNP on a specific gene that affects a catalytically active residue with a functional impact on the protein, so if it messes up the protein, then you could have something like a predisposition toward cancer. So several years ago, we developed an algorithm and a program to search for SNPs on single genes, so the genes that we're interested in, and then we have to do statistics to figure out if these mutations are really relevant. We call this hypothesis-driven SNP search or hidden SNPs. And then we use these other techniques I mentioned before for molecular simulations to figure out if these mutations affect how the protein works or how it moves. So the structure, the function, or the dynamics. In general, when people are looking for these mutations on DNA, what they do is they use what are called genome-wide association studies. I'm not going to go into the details, but what these genome-wide association studies, or GWAS for short, result in are huge databases that have all of the SNPs of people that have a disease and people that don't have a disease, so that you can compare. So we can use those databases to search for our SNPs. So we start at the comparison stage, and then we search only in the genes that affect the enzymes that we're interested in. One of the enzymes, the one that I told you Pavel and Alice investigated, is called AlkB. AlkB is in charge of fixing damaged nucleobases. So when DNA has certain damage, this enzyme fixes it. It can fix several different things, including single-stranded DNA, double-stranded DNA, etc. And the enzymes are divided into families. The AlkB family has one enzyme in bacteria, and there are 9 homologs, so 9 enzymes that are related to AlkB in humans. So what we were interested in is, are there any relations between cancer and specific enzymes in AlkB? And so what Pavel did is he did a search of all prostate cancer SNPs on AlkB enzymes, and what he found is that indeed there is a cancer mutation that changes one specific amino acid in a protein, in this protein called AlkBH7, that is related to prostate cancer. And then what Alice did is she followed up this prediction by figuring out what this mutation does. So this is a comparison of what we call the wild type enzyme, that's in gray, and the mutant, or the cancer mutant enzyme, in yellow. And as you can see, they're pretty similar. However, there are important differences. One is over here in this loop, and another one is in this region over here. This region is what in the enzymes are called the active site. This is where the action happens. And so when you have the cancer mutation, this affects the protein where the action happens. So what Alice followed up on was trying to figure out, does that really affect how the enzyme functions? One of the things that the enzyme has to do is it has to accept, or has to bind certain chemicals to do a reaction. And what happens when you have the cancer mutation is that these chemicals, or these molecules, are not bound. Whereas if you have the regular enzyme, the wild type enzyme, they're always bound in the active site. So she was able to predict that because of this cancer mutation, the enzyme would not be able to bind the required molecules to do the reaction. This was confirmed by our experimental collaborators at Michigan State University, Professor Hausinger and Dr. Mueller, where they found that indeed Alice's prediction is correct. So the wild type enzyme does bind, and the cancer mutant does not bind the substrate. And so our hypothesis is, because the enzyme now cannot bind the molecules that it requires in the cancer variant, then it cannot fix the DNA, and this leads to mutations which lead to cancer. Another example that we have done is following up on what other work that Pavel did, where he generated what we call the DNA repair cancer database. And so as you can see here, we have many different mutations in many different enzymes and in many different types of cancer. So we focused on only one right now, ApoBec3H. ApoBec3H is a member of a very important family of enzymes called cytidine deaminases. And these are very important enzymes because they help us fight viruses like HIV. We uncovered a missense mutation that results in a change that is related to lung cancer. And so what a student in my group, Mark Hicks, figured out is that this mutation that is located at position 121, what it does is it basically creates creates significant changes in the region where the mutation is, that effectively lock up the enzyme. By locking up the enzyme, that changes the effects of several different structure and dynamical features, including these things that keep the proteins together called hydrogen bonds. And so the cancer mutant has a huge change in hydrogen bonding, whereas some other mutants that are known to not affect so much, indeed don't affect so much. Mark also predicted that there would be a second mutation that can be done that would restore this, what's called the wild type activity. And so once we had these two predictions, that the cancer mutant affects the structure and the function of the enzyme, and there is a possible second mutation that can be done to rescue, we contacted another collaborator in Canada, Professor Linda Chelico and her lab, and they did a huge number of experiments which showed that indeed Mark's predictions are correct. So the cancer mutant is not able to be expressed, so it cannot form. Because it cannot form, it cannot do its activity. The recovery mutation definitely does express, and it does work, and also it dimerases. So now we're 2 for 2 with these cancer mutations, and they're predicting what they do, and getting experimental confirmation from different collaborators, and we collaborate with people literally all over the world, and able to enable these studies and other studies. Finally, these results, I'm not going to go through all of them in the interest of time, but what they were able to do is they were able to use human cells that are healthy and human cells that have lung cancer, so lung cancer cells and healthy and lung cancer cells, and they were able to show that the reason why this mutation is related to lung cancer is because you have a lot more damage when you have this enzyme. So again, I'd like to thank my students and collaborators and previous students who have done all of this work and funding from these different funding agencies, computing time from these agencies, and I welcome you to UNT. Thank you very much.